In this video, you will learn how to perform a mini prep. Bacteria-containing plasmids will be grown in liquid culture. The plasmids will then be extracted using alkaline lysis method. Finally, column chromatography will be performed to purify the plasmid DNA. When plasmid DNA is purified on a small scale, as performed in this activity, it is called a mini prep. Sterilize an inoculation loop, then using aseptic technique, transfer a single colony of bacteria from a petri plate to a properly labeled culture tube with nutrient broth. When finished, recap the tube and sterilize the inoculation loop. Place the culture tube in a shaking incubator, shaking water bath, or tube roller in an incubator. Grow the culture overnight at 37 degrees Celsius. After an overnight incubation, the culture should appear cloudy. Transfer 1.5 milliliters of the culture into each of two properly labeled microcentrifuge tubes. Place the tubes in the microcentrifuge in a balanced pattern. Put the hinges of the tubes facing outward so that the pellet will be easy to locate after centrifugation. Centrifuge the tubes containing cultures for 1 minute at top speed to pellet the bacteria. When the microcentrifuge is stopped, remove the microtubes. Locate the bacterial pellet. It should be below the hinge of the tube. Remove the liquid or supernatant from each tube using a micropipette or a vacuum source. Avoid touching the pellet. When finished, discard the supernatant. Using a fresh tip each time, pipette 250 microliters of resuspension solution into each tube. Repeat this step, adding 250 microliters of resuspension solution to the second tube. Resuspend the pellets by vortexing or flicking the bottom of the microcentrifuge tube to break up any clumps of bacteria. Pipette 250 microliters of lysis solution into each tube. Mix both tubes by gently inverting 6 to 8 times. Do not pipette or vortex. Pipette 350 microliters of neutralization solution into each tube. Mix by gently inverting 6 to 8 times. A precipitate should form. Spin the tubes in a microcentrifuge for 5 minutes at top speed. Place a green mini column into each of 2 capless collection tubes. Remove your samples from the microcentrifuge. Using a pipette, remove the supernatant from the centrifuge tube, being careful not to disturb the pellet. Notice that only the pellet is left in the microcentrifuge tube. Now transfer the supernatant that you removed into a green mini column. Repeat this procedure with your other sample and transfer the supernatant into a second green mini column. Place both green mini columns into the microcentrifuge in a balanced pattern. Spin the columns in the capless collection tubes in the microcentrifuge for 1 minute at top speed. Take the columns out of the capless collection tube. Discard the liquid flow-through and replace the column into the collection tube. Do this with both columns. Pipette 750 microliters of wash solution into each column. Place both columns into a microcentrifuge in a balanced pattern. Spin the columns in the capless collection tubes for 1 minute at top speed. Discard the flow-through from each collection tube. Place the columns back into the collection tubes and spin for 1 minute to dry out the columns. Discard the capless collection tubes and place the columns into clean microcentrifuge tubes. Pipette 50 microliters of elution solution directly onto the column bed of each column. Allow the elution solution to absorb into the column for 1 minute. Spin the columns in the clean microcentrifuge tubes for 1 minute at top speed. Since the caps are not closed, the microcentrifuge may need to be run without closing the inner lid. Discard the columns and close the caps on the microcentrifuge tubes.